Good afternoon. In today's session, I want us to discuss the thymus. And as usual, my name is Maurice Munir, and I'm the redactor. So, what will be the scope of 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 this lecture? One, we discuss the gross anatomy of the thymus. That won't take up much of the part. Then, secondly, discuss how it develops. Then, this is what I focus on majorly: the histology of the thymus. And then we discuss some immunology, um, which relates to the thymic education. So let's start with the gross anatomy and embryology. So the thymus is a structure found in the anterior and also in the superior mediastinum. So, and it has two lobes. Okay. So the thymus develops from the third pharyngeal pouch. Okay. The third pharyngeal pouch. Also from the third pharyngeal pouch, you get the inferior parathyroid. Okay. So the mesoderm of the third pharyngeal pouch is actually what uh, um, um, gives rise to those the epithelial reticular cells, which um, constitute the the supporting framework of the thymus. Then the thymocytes, these immature T cells actually migrate from bone marrow or bone marrow precursor and invade these developing thymus. Okay, so thymus continues to grow till puberty. So at puberty, that is when it's in its maximum size. Then eventually it undergoes atrophy with infiltration of adipocytes. So adipocytes infiltrate this thymus, but in an adult, still it's functional. Okay, it's functional, but that tells you that the thymus plays a very important role in during childhood, but the role tends to regress as one ages. So if you have the move of the gland or the thymus in, during childhood, that would affect the individual more than if the same was removed when one is an adult. Okay, so this is the thymus. Okay, you can see this is the fibrous pericardium. So within the superior mediastinum, we have this thymus here. Okay, the thymus. Um, it is drained by a vein known as the thymic vein, which drains majorly to the left brachiocephalic vein. Okay, left brachiocephalic vein. Okay, so from the third pharyngeal pouch, you have co-development of both thymus and parathyroid, and then they separate. Okay, they separate, and the thymus migrates caudally to the superior mediastinum to form this bilobed structure known as the matured thymus. Now, failure of this separation causes now both of these to be located in the superior mediastinum. So that is why if these two don't separate, there is a possibility that you find some inferior thyroid, or rather inferior parathyroid, sorry, the inferior parathyroid glands may be found within the superior mediastinum. Then down here is what we'll be discussing under thymic education. So this is simply a flow chart which tries to describe the histology of the thymus. So it has a capsule or the stroma and the parenchyma. Now, the capsule is simply a thin connective tissue which surrounds the thymus. Okay, then it sends in some septi or trabeculae to lobulate the 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 lobes into incomplete lobules. So it's a bilobed structure. Then these two lobes are further divided by septa or trabeculations to form incomplete lobules. Okay. Then the parenchyma, on the other hand, has a cortex and a medulla. Now the cortex, it is darkly stained, okay, because it contains closely packed small T lymphocytes or the T cells. So the T cells are small and closely packed within the cortex, as opposed to the in the medulla where it is lightly stained because the T cells are larger in size and they are loosely packed. Now, the distinctive feature of the medulla is presence of these thymic corpuscles or the Herschel's corpuscles, and these are simply hyaline material which are surrounded concentrically by type 6 epithelial reticular cells, okay? The type 6 epithelial reticular cells. Then we have now the epithelial reticular cells. There are 6 types. One, the function major is to one, the epithelial reticular cell type 1 forms the blood thymic barrier. But also what I need you to know about epithelial reticular cells is that they are also known as the nurse cells. The nurse cells, they participate in thymic education. Okay? So consider the thymus as a military camp where now these militia, now the T cells are trained how to identify an enemy. So an enemy is a foreign antigen and take action on them versus how not to identify uh, a fellow comrade, and hence not in quotes kill them. So the thymus is like a military camp where now these militia or the thesis now undergo training. Okay, and now the trainers are the epithelial reticular cells. Okay, so the function of the thymus is simply to produce mature T lymphocytes, and also they secrete. It is considered as part of the endocrine system because it secretes the foreign hormones, thymosin and thymopoietin. Okay? So here is the capsule. You can see the capsule. Okay, and then you can see the capsule is sending in some septa or this trabeculae to now lobulate the thymus. Okay, so these septi are important because it is through these septa that efferent lymphatics uh, move out of the gland. So as opposed to the lymph nodes have afferent and efferent lymphatics. The thymus, on the other hand, has only efferent lymphatics. Also, blood vessels and nerves um, are access the gland via these septi. Okay. So you can see this is a lobule, this is one lobule. Then if you focus on that one lobule, you can see it has an outer darker staining part and inner lighter staining part. Okay? So this darker outer staining part is the cortex. Okay? So this is the cortex. Okay? That's the cortex. And then the lighter staining part, that's the medulla. Then you can see the corticomedullary junction. Okay? It's not well defined, but you can make out that this is the medulla and that's the cortex. Now we have some various, these dark staining, those are some small T cells, okay? So within the cortex, two types of selections occur. One is known as the beta selection, and the other one is known as the positive selection. We discuss that later on. So what are some various cell types which you expect to find in the cortex? One, we have the epithelial reticular cells from type 1 to type 3. So epithelial reticular cell type 1, type 2, type 3 are found within the cortex, okay? And the way you can distinguish them from these with dark staining nucleus, those are the T cells or the T lymphocytes, okay? But these ones which with a light staining nucleus, you can see this euchromatic nucleus here with a prominent nucleus, those are now the epithelial reticular cells, okay? Now, on the matter of identifying between type 1, type 2, type 3, that would call for special uh, histochemical stains to distinguish that, okay? So these are the epithelial reticular cells from type 1 to type 3 are found within the cortex, okay? Now, which other cell types are found within the cortex? You also have macrophages. Now, this is a macrophage there, okay? And this macrophage, specifically here, you can see it has phagocytosed some cells, some T cells. So the function of the macrophages is to phagocytose these apoptotic T cells. So within uh, um, the cortex, those cells which uh, uh, um, there is this selection, and if a cell does not qualify for the criteria of selection, it undergoes apoptosis, and then the debris is cleared by the macrophages. So that there is a macrophage, okay? And but this here, you can see this is an epithelial reticular cell, okay? You can see how the euchromatic nucleus with a prominent nucleus over there. And then these other ones here, those are the T cells. So that's a macrophage there. Then the medulla, on the other hand, you can see it is slightly staining because it has those larger T cells. But then the distinctive feature is now these bodies, or corpuscles. These are the thymic corpuscles or the Herschel's corpuscles. And these are simply, uh, you have keratin at the center, and you can see you have some concentric cells surrounding that keratin material. Those are the type 6 epithelial reticular cells. So the cell types found within um, the medulla will be now the type 4, 5, 6 epithelial reticular cells. You also have the T lymphocytes, okay, the T lymphocytes. You also have some macrophages within the medulla to clear those cells which do not abide to the criteria of negative uh, selection. Now, these are now the thymic corpuscles. They are simply some small rounded or ovoid structure present in the medulla. They consist of that hyaline mass at the center, which is surrounded by the type 6 epithelial reticular cells. They actually increase with age, okay, and they are a representation of degenerating thymocytes. So if you take a thymus of a young individual and observe uh, the, the thymic medulla, the number of the corpuscles will be lesser than that of an adult. Okay, then these type 6 epithelial reticular cells, they secrete the following interleukins, interleukin 4 and 6, and these interleukins are very important for maturation of the democytes. Now, these democytes are those T cells within the medulla. Okay, and they also, now it is the thymic corpuscles which actually produce the thymosin and the thymopoietin hormones. So this is a medulla. You can see we have those bodies. You can see eosinophilic hyaline mass, that's keratin there, and surrounded concentrically by the type 6 epithelial reticular cells. Okay, and you can see some lymphocytes, those darkly staining cells. Okay, so just as a recap, cortex, darker, more densely packed, small lymphocytes, medulla, inner, lightly staining, less larger sized T lymphocytes. Okay, and then but the distinctive feature or characteristic of the medulla is the presence of those Herschel's corpuscles. Then also within the medulla, you find what are known as post-capillary venues. Okay, so now this is a Herschel corpuscle. You can see that eosinophilic hyaline mass, and then it is surrounded by those type 6 epithelial reticular cells. But then you can see that one there, that's a venule, okay, a post-capillary venule. Yes, once these T cells have undergone training, they give rise to what are known as naive T cells, mature naive T cell. Now that cell will exit the medulla via these postcapillary venules, and now it will go and seed peripheral lymphoid organs such as the spleen and the lymph nodes. In the lymph nodes, they form the paracortex. In the spleen, they form the periarterial lymphatic shield. So that is the histology of the thymus. Okay, so let us talk about these epithelial reticular cells, okay? So why the name epithelial reticular? So they have both characteristics of epithelial cells and reticular cells, okay? And these are the cells which are now derived from the third pharyngeal pouch. They are also known as the NAS cells because they usually provide a required microenvironment for the differentiation and the maturation of these cells, okay? So these cells, NAS in quotes, like NAS the uh, um, the naive T cells, okay? the naive T cells. So type 1, okay, so type 1 epithelial reticular cells, they simply are important in the formation of the blood thymic barrier. Just remember that. Okay? Formation of the blood thymic barrier. Then type 2, they usually create an isolated compartment in the cortex, and they educate the T cells within the cortex. Then type 3, they are the one which form the functional barrier between the cortex and the medulla. Okay, so they are the cells which now form the corticomedullary junction. It's a functional barrier rather than an anatomical barrier. Okay, it's a functional barrier. Then type 4, these ones are now found within the medulla, and they also participate in the formation of that functional barrier, the corticomedullary junction. Now, type 5, on the other hand, they usually form a cellular framework in the medulla, okay? They form a cellular framework in the medulla. And type 6, they are the ones which I showed you, forming those concentric layers in the habeas corpus, and they secrete the interleukins 4 and 6, and those ones we said are important for thymocyte maturation and also produce thymosin and thymopoietin hormones, okay? So, type 1, please remember type 1, formation of the blood thymus barrier. Type 6, the habeas corpus. Type 3 and 4, formation of the functional corticomedullary barrier, that functional barrier between the cortex and the medulla. Let's discuss about this blood thymic barrier, and maybe later on we will see the function of this barrier. Now, you have a capillary in here in the thymus, within the cortex of the thymus. So that capillary is separated from the thymic cortex by a barrier. Now, the importance of that barrier is big. You don't want these immature T lymphocytes to be exposed to foreign antigen before they are trained, or rather, be exposed to antigen. Let me use the term antigen before they are trained on how to identify these self versus foreign antigens. Okay, so you have to form a barrier. Now that barrier is formed by the following components. One, the endothelium. Okay, so the capillary endothelium. Okay, and then its basal lamina. So the basal lamina, which is formed by the pericytes. Okay, and then the perivascular connective tissue. Then within the perivascular connective tissue, you might occasionally find a macrophage. So macrophages may be in there. And then next will be now the type 1 epithelial reticular cells. Okay, before you get to those type 1, you have the basal lamina of the type 1 epithelial reticular cell. Okay, the basal lamina of type 1 epithelial reticular cell, and then the epithelial reticular cell type 1, the type 1. Okay, so remember type 1 epithelial reticular cell is important for the formation of the blood thymic barrier. So the blood thymic barrier is formed by the capillary wall, and this wall will be formed by the endothelial cell and its basal lamina. Okay, and then the perivascular connective tissue, and within it you might find macrophages and also fibroblasts, and then type 1 epithelial reticular cell together with its basal lamina. So we have tight junction between these epithelial reticular cells, and that those tight junctions are important for the integrity of the blood thymic barrier. So this is simply a longitudinal section to show you that. So this is the lumen of that capillary, and then you can see that there is the endothelial cell, but then this is the thymic cortex. So this barrier here will be formed by now the capillary wall. Then in between the two, here will be the connective tissue. But then type 1 epithelial reticular cells will be there as the outermost component of the blood thymic barrier, and their tight junctions are important for the integrity of that barrier. So thymic education. So we have 3 checkpoint selections. So the first selection and the second selection, beta selection and the positive selection, those two occur within the cortex. But then negative selection occur within the medulla. So what is beta selection? So beta selection simply select cells which express a pre-T cell receptor. So a T cell receptor is made up of an alpha chain and a beta chain. So you have a T cell precursor which has come from the bone marrow. So this T cell precursor has come from the bone marrow. T cell does not express a T cell receptor. It does not. Okay, nor does it express the cluster of differentiation molecules, the CD molecules. It does not. Okay, but it has to express these molecules for it to mature. So beta selection is now this T cell precursor cell undergoes the double negative stages. So double negative means uh, the cell does not express the CD4 and the CD8 molecule. So it is CD4 negative and CD8 negative. Those are the double negative stages, okay? And there are four of them, DN1, 2, 3, and 4, okay? But in those double negative stages, a B chain is formed, okay? A B chain is being formed, and this B chain uh, um, uh, binds to an invariant alpha chain. So in the double negative stages, a B chain is being formed, and this B chain binds to an invariant alpha chain. Now, when that happens, you form as a double negative T cell. So it is CD8 negative, CD4 negative still. So it is double negative still. No CD8 or CD4 molecules have been expressed. But it has a pre T cell receptor. Okay, this pre T cell receptor has an invariant alpha chain, but the beta chain locus is functional. Okay, so the double negative stage is involved with the formation of a beta chain for the T cell receptor. But on the other hand, now when a pre T cell receptor is formed, uh, T cells are signaled to proliferate, and as they proliferate, they express both CD4 and CD8 molecules, and these two molecules are expressed within the same cell, and now it ceases to become a double negative, it becomes a double positive T cell, and now these double positive T cells are involved in alpha chain selection, okay? And once that happens, a cell with a functional T cell receptor is formed, okay? But this cell with a functional T cell receptor um, still is double positive, and now it proceeds to the inner part, um, of the cortex, where now thymic positive selection will take place. Now let's discuss this thymic positive selection. Now we have this double positive T cell. Now, first of all, this positive selection, it's important in that it selects cells that are able to interact adequately or appropriately with self MHC or the HLA molecules, so the major histocompatibility molecules and the human leukemia antigen molecules. Okay, so we have these double positives, so it has both CD8 and CD4 molecules. Okay, so so we have these thymic epithelial reticular cells. So these most likely are the type 2, the type 2 within the cortex. So these double positive T cell, its fate is determined during this positive selection. So if this double positive T cell interacts with the MHC type 1 molecule expressed by the thymic epithelial reticular cells, it will now become a CD8 positive. It will upregulate the CD8 molecules and downregulate the CD4. And so it will become CD8 positive, CD4 negative T cell. But if the same happens, when it interacts with the MHC type 2, it will upregulate the CD4 molecules and downregulate the CD8, and it will become CD4 positive, CD8 negative T cell. Now these two cross the corticomedullary barrier and head to the medulla for negative selection. Now, on the other hand, the double positive T cells, which now inappropriately interact with the thymic epithelial reticular cells, that is, they do not bind to the MHC molecules, or they bind too strongly. Those cells do not receive an enough survival signal, and they undergo apoptosis, and they are cleared by the macrophages, the cortical macrophages of the uh, thymus. Okay, but then within the medulla now, negative selection on the other hand, sorry for that. So negative selection on the other hand select T cells that do not recognize self-antigens, and this concept is known as central tolerance. Okay, central tolerance, very important. Now, how is that happening? In the medulla, you have what are known as medulla thymic epithelial reticular cells, or thymic dendritic cells. Now, classically, you know that it is MHC type 1 molecules. MHC type 1 are the ones which are able to uh, express self-antigen. But within the medulla, the thymic medulla, that law is not obeyed, in that MHC type 2 molecules also express self-antigens. And how does that happen? It is because the thymic dendritic cells phagocytose these medullary thymic epithelial reticular cells, and they express now the self-antigens of these cells as self-antigens using now MHC type 2, okay? And now we have these cells, take this cell, CD4 positive, CD8 negative. So CD4 positive cells interact with the MHC type 2. Now, if this cell recognizes this self-antigen, so there is a strong interaction, you don't want that cell to be released to the body, because now this cell would recognize self-antigens and surmount an immune reaction against self-antigens. That would now lead to autoimmune diseases or disorders. So that cell is undergoes apoptosis. Okay, but then cells which now do not interact with the self-antigens, or they do so but with very low affinity, those cells receive now the sufficient survival signals, and they form now the mature naive CD4 positive T cells, and now they exit the thymus and go to seed the peripheral lymphoid organs. Okay, so this was the case for CD4. Now CD8 will be just the same. So if it was CD8 positive here, it would interact with MHC type 1. If there was a strong interaction, the same fate. If there was no interaction of low affinity interaction, the same fate would occur. Okay, and that, my friends, is the thymic education in a nutshell. So thank you very much for your attention, and do subscribe for more anatomy and very uh, very much immunology. I also have pathology videos. So subscribe for more content. Thank you very much.